Hello everybody. Today I'll be discussing the purine catabolism. The degradation of nucleic acids occur in the intestines. Now, what are all the enzymes involved in the degradation of nucleotides? The first is the pancreatic nucleases, which hydrolyzes the polynucleotides into oligonucleotides. Then the phosphodiesterases convert them into mononucleotides. At the intestinal mucosal surface, the nucleotidases will then convert these nucleotides into nucleosides. The nucleosides which are released are then transported to the enterocytes. Here the nucleosides are further degraded by nucleosidases to free bases and ribose-1 phosphate. The dietary free bases are usually degraded to uric acid. So in this slide, we can see what happens to the nucleic acids. The nucleic acids, as you can see, are first converted to the nucleotides. Then after that, they are acted upon by the nucleotidases to the nucleosides. The nucleosides then are acted upon by the nucleoside phosphorylase and converted to free bases and the ribose-1 phosphate. Some of the bases are then reused to form nucleotides by the salvage pathway, whereas the others get degraded to uric acid, which is the end product of purine catabolism. Now just let's look at the catabolism of purines. Now firstly we'll look at what happens to adenosine. Adenosine monophosphate, there are two pathways which are available for it. It can be first deaminated by AMP deaminase to form IMP, and then it is acted upon by a nucleotidase to be converted to inosine. The other pathway which is open to it is that it is first acted upon by a 5' nucleotidase to form adenosine and then deaminated by adenosine deaminase to form inosine. Once inosine is formed, then it is acted upon by a purine nucleoside phosphorylase to form hypoxanthine. And this hypoxanthine, with the action of the enzyme xanthine oxidase, is converted to xanthine, and xanthine, again by the action of the enzyme xanthine oxidase, is converted to uric acid. Now, what is the pathway for guanosine monophosphate? Guanosine monophosphate is first reacted by 5' nucleotidase to form guanosine. Guanosine is then acted upon by the nucleoside phosphorylase to form guanine. Guanine is acted upon by an enzyme guanase to form xanthine, and xanthine is then acted upon by xanthine oxidase to form uric acid. So the two purines, adenosine monophosphate and guanosine monophosphate, are finally converted to uric acid. So please note the important enzymes in this pathway. Xanthine oxidase, which is the site where we can have the inhibition occurring, and the drug allopurinol acts at this site, and also we have adenosine deaminase, the deficiency of which causes a very important disorder known as ADA deficiency, which we will be discussing at a later stage. Now, where is the site of formation of uric acid? It is in the liver. The quantity of uric acid which is present in body water is known as the miscible pool, and it comes to around 1130 milligrams. The plasma contains a higher concentration. What do you mean by turnover? Turnover is the rate at which the uric acid is synthesized and how it is lost from the body. What is the normal level of uric acid? It is 3 to 7 milligram per deciliter in males and 2 to 5 milligram per deciliter in females. The daily excretion is 500 to 700 milligram per day. A source which is rich in uric acid is organ meat, and source which is low in uric acid is milk, egg, and cheese. So let's look at the uric acid metabolism. You have what is known as the urate pool, and the factors contributing to an increase in the urate pool is the dietary intake. Secondly is the tissue breakdown of nucleic acid, and thirdly it is the endogenous synthesis of purines. Now what goes out of this urate pool is how much is moves out from this urate pool depends upon the excretion in the gut and also the renal excretion. Now, what causes hyperuricemia? It is either the overproduction of uric acid or the under-excretion of uric acid. Both these are contributory factors which can result in hyperuricemia. Hyperuricemia, if it is only marginally high, is asymptomatic. But when we have high levels of uric acid, then we find that the patient will have a disorder which is known as gout. And the urate crystals can accumulate and cause, these crystals can result in what is known as tophi in the joints. So it is known as chronic tophaceous gout. It can also result in renal manifestations. So let's look at the disorders of uric acid catabolism. Hyperuricemia. Hyperuricemia means the elevation of uric acid in the blood, and the serum uric acid levels will be more than 7 milligram per deciliter. There would also be excretion of uric acid when it is known as uricosuriia. Now, what are the primary causes for hyperuricemia? The primary causes could be due to certain enzyme deficiencies. The first enzyme is the 5-phosphoribosyl amido transferase. Now, as you know, this is a regulatory enzyme, and this is the step at which we can have regulation of purine biosynthesis. So if there is an inherited abnormality in this enzyme, that is the 5-phosphoribosyl amido transferase enzyme, it could affect purine metabolism. Now what we see in this case is that the enzyme is active, but it is not sensitive to feedback inhibition by its products, that is either inosine monophosphate, guanosine monophosphate, or adenosine monophosphate, and this can lead to overproduction of purine nucleotides. The second enzyme which can be affected is the phosphoribosyl pyrophosphate synthetase, or the PRPP synthetase. Now, certain mutations have been seen which are X-linked, and these have been identified in this enzyme. And because of these mutations, it is not subjected to the normal allosteric control, and this would lead to an increased production of PRPP, which in turn would lead to an increased production of the purine nucleotides, which in turn can result in increased production of uric acid. The purine overproduction could also be due to the deficiency of the enzymes HTPRTase and APRTase, which, as you know, are very important enzymes of the salvage pathway. Deficiencies of these enzymes can result in what is known as the Lesch-Nahen syndrome. So there is decreased salvage of hypoxanthine and guanine bases, and because of that there will be increased availability of PRPP. This, and in addition to this, since the bases the purines levels are decreased, there would be decreased feedback inhibition. Next, we can have glucose 6-phosphatase deficiency, like we see in von Gerke's disease, that is the glycogen storage disorder type 1. In this, when glucose 6-phosphatase is deficient, what happens? Glucose 6-phosphate is not converted to glucose. So what happens to this glucose 6-phosphate? It gets channeled through the HMP shunt, and we have increased production of ribose phosphate. When there's increased production of ribose phosphate, there will be an increased synthesis of phosphoribosyl pyrophosphate. Lastly, we could have a variant of the enzyme glutathione reductase. Again, when there is a variant of this enzyme, we find that there is an increased production of ribose phosphate. With this, we have increased production of phosphoribosal pyrophosphate, and because of that there will be increased synthesis of purines, and because there is increased synthesis of purines, there will be increased degradation of these purines to uric acid. Now, what is secondary hyperuricemia? The secondary hyperuricemia could be due to cancer or rapidly growing malignant cells like leukemias or lymphomas because of increased tissue breakdown after treatment of malignant tumors or increased tissue damage due to trauma. So secondary hyperuricemia could be either due to cancer, where we have rapidly growing malignant cells, and this is particularly seen in cancers like leukemias and or lymphomas. It is also seen when there is increased tissue breakdown, particularly after chemotherapy, or increased tissue damage due to trauma. Now, the next reason for secondary hyperuricemia could be the under-excretion of uric acid, and this could be due to either renal failure, or there could be increased or elevation of lactic acidosis due to uh, the renal threshold for urates being affected, leading to elevated total body urates. Or it could be due to the use of thiazide diuretics, which inhibits the tubular secretion of uric acid. So the three things to remember about under-secretion of uric acid is the renal failure. Secondly, elevation in lactic acid. This is because the renal threshold for urates is affected, and this elevates the total body urates. And thirdly, the use of thiazide diuretics, which inhibits the tubular secretion of uric acid. Now, the important disorder associated with hyperuricemia is gout. Gout is characterized by high levels of uric acid, and as you know, uric acid is the end product of purine catabolism. So, as I've already told you before, hyperuricemia may be either due to overproduction of uric acid or the under-excretion of uric acid. Now, what happens? This leads to the deposition of monosodium crystals of urate in the joints. The deposition of these crystals, the urate crystals, results in an inflammatory response. And this results in what is known as an acute and then chronic gouty arthritis. The nodular masses of monosodium urate crystals or tophi may be deposited either in soft tissue or when it is known as chronic tophaceous gout. We could also have the accumulation of these uric acid crystals in the kidney, which are known as urate stones. Also, the deposition of urate crystals in the renal medulla, which is also otherwise known as urolithiasis, can cause renal damage. Now, gouty attacks may be precipitated by a high purine diet and increased intake of alcohol. Increased intake of alcohol can lead to increased accumulation of lactic acid. So, one important thing to remember is, in such cases, is a no to alcohol. Tophi. I told you tophi is a nodular mass of monosodium urate crystals. This initiates an inflammatory response which causes infiltration of the granulocytes, and there is a tendency to phagocytose the urate crystals. This releases certain oxygen metabolites which can damage the tissue and release lysosomal enzymes, and this ultimately results in the inflammatory response that we see in the case of arthritis. What is the treatment for gout? The palliative treatment is bed rest, a purine-free diet, and of course, very important, restrict alcohol consumption. The drugs which we can give can be the anti-inflammatory drugs like colchicine, which decreases the movement of granulocytes into the affected areas. Then we can give the non-steroidal anti-inflammatory drugs like endomethacin or diclofenac, which inhibits the synthesis of prostaglandins, which mediates the inflammatory response, or simple aspirin, which can provide pain relief. Now, the specific treatment is based on either increasing the renal excretion of uric acid through uricosuric drugs, decrease the synthesis of uric acid using an inhibitor, or increase the oxidation of uric acid. And the drug used is urate oxidase. The uricosuric drugs enhances the renal excretion of uric acid. So they are the specific inhibitors of tubular reabsorption. And the therapeutic strategy is to lower the uric acid levels to below the saturation point, which will prevent the deposition of the urate crystals. Some examples of uricosuric drugs are the probenizids, the sulfenpyrrazone, halofenate, and salicylates. All of you are familiar with the drug allopurinol, which acts as an enzyme inhibitor of xanthine oxidase. Allopurinol is a structural analog of hypoxanthine and a competitive and suicide inhibitor of xanthine oxidase. The accumulation of hypoxanthine and xanthine makes it more soluble when we use this inhibitor allopurinol, and there would be a rapid fall in uric acid. Xanthine oxidase converts the allopurinol to alloxanthine, which is a more effective inhibitor of xanthine oxidase and an example of suicide inhibitor. So if allopurinol is administered, then the production of uric acid is decreased and we have more of hypoxanthine and xanthine being formed, and as they are more soluble, it will be excreted out in the urine. Hyperuricemia. This relates to decreased uric acid levels in the serum, which is less than 2 mg per deciliter. And it's mostly associated with a rare genetic defect, firstly in the enzyme xanthine oxidase. Leads to the increased excretion of xanthine and hypoxanthine, and xanthinuria frequently causes the formation of xanthine stones in the urinary tract. Now, a very important disorder associated with hyperuricemia is the adenosine deaminase deficiency. Deficiency of ADA causes a severe combined immunodeficiency, SCID, which is involving both the T cells and the B cells. ADA deficiency results in the accumulation of deoxy-ATP, which becomes an inhibitor of ribonucleotide reductase, and it will inhibit DNA synthesis and cell replication. Now, adenosine deaminase is expressed in the cytosol of cells. The human lymphocytes have the highest activity. Deficiency of ADA will cause accumulation of adenosine, which is then converted to nucleoside triphosphate and deoxynucleoside triphosphate. As DATP would increase, the ribonucleotide reductase gets inhibited and prevents the formation of all the deoxynucleotides, phosphates, DNTPs. It will prevent the formation of all DNTPs, and therefore the cell will not be able to make DNA. It is an autosomal recessive disease, and I told you it causes severe immunodeficiency of both the T and the B cells. The children are kept in the sterile environment and they usually die by the age of 2. In the United States, the ADA deficiency accounts for almost 14% of SCID or severe combined immunodeficiency. The treatment is gene replacement therapy. You are all familiar with Ashanti D. Silver, In 1990, she became the first patient to receive gene therapy for ADA deficiency. So here we see her at the age of 13, and she continues to lead a very healthy and active life. The two fathers of human gene therapy is Francis Collins and French Anderson. Here you can see French Anderson along with Ashanti De Silva as she was undergoing treatment for ADA deficiency.